So good evening all of you and welcome back to Excelsior Academy. In today's topic, we are going to study about action potential, or in other words about the membrane potential. Okay, so what is action potential? Action potential is the mode through which a neuron transports signals. So that here we are talking about the neuronal signal, right? Electrical signal. So till now what we have discussed is that at the synaptic cleft, we talked about the ligand gated channels, right? We talked about the ion channels. Now, all these channels and the ligand gated channels are helping in transmission of impulse from one neuron to the another neuron. And this is how your signal or the impulse, electrical impulse, is transferred from one neuron to another. Now, I'll give you one small example which relates to all of us. So when we talk about memory, like we want to study and we are not able to remember stuff. So exactly what is happening that time is, see, when you revise, okay, when you revise your topics again and again, it means what? You are sending the same signal again and again to your brain, right? And that time, sending your same signal for long time, what happens? It gets converted into your long-term memory, okay? And that's why when you understand, you don't forget. But if you go only and you just study only once, suppose you just heard me today and you just listen, and after that you are not revising, what happens? This entire thing what you have learned today will go into a short memory. Okay, so this is like erasable. It means that when something else will come into your mind, and in order to make that and into a long-term memory, whatever was there for your short term, it will get erased. Okay, and then that space will be taken by the long-term memory. So this is what happens, and then you don't remember. So even if you try hard, array, I have studied this thing once, okay, ma'am told this thing and all, but I'm not able to remember. Why? Because your neurons were not getting the same signal. So once you give the same signal every time, then only the revision happens and then it becomes your long-term memory. Okay. So this is just for the understanding information. So what is action potential? It is mode through which a neuron transports electrical signals. So it means that if you have one cell over here, another cell over here, so they are just, nearby, and from here to here, the signal is transferred. Okay. It is defined as brief change in the voltage across the membrane. Yes, due to the flow of certain ions into and out of the neuron. So in case like in our house also, whenever current is there, what is that? It is nothing but the flow of electrons, right? In the same way, in the case of action potential also, in the membrane, it is the flow of certain ions. Either they are going intracellular or extracellular. So this flow is actually maintaining the action potential. Okay. So now we know that after studying for past 2 weeks, now we know that it is because of your Na plus K plus ATPase channel. Remember? So this comes under your active transport. Now, in this channel, which ion comes and binds first? Which one is the first one? Sandhya, which ions comes and bind first?
Sodium ion.
Yes, very good. So it is the sodium. So first 3 Na plus comes and binds, and then 2K plus with ATP hydrolysis. Okay. So now we are going to see how this active transport of Na plus K plus ATPase is maintaining or regulating the action potential. Okay. Okay. So now resting membrane potential. So what is the resting membrane potential? So we know it is around minus 70 millivolts.
-70 MV.
Yes. So, but see, so entire brain is there. Your neurons are also in connection with the motor neurons. Muscular junctions are there, right? So each neuronal activity will vary. So the generally it has given that it is between the -50 to -70 millivolt. Okay, now how do you know about that the resting potential is around -70 millivolt? So there is a technique called patch clamp. Okay, so it's a patch clamp technique by which it was understood and measured that the resting potential of the membrane is near about -70 millivolt. Okay, so only this technique is left, which we can do tomorrow or on Monday, so not a big thing. Okay, so the value depends on the types of ion channels that are open and the concentration of different ions at the intra and the extra fluids. Okay, so every time you don't get your neuronal cells to have the resting potential of -70. See, because you are working, you are studying or doing some activities, so always your neurons are, passing the impulses. That is only when at rest, that is when you're not sending any signal, you get your -70 millivolt as the resting potential. Okay, now it is not only that the channel are open or closed, it is also the difference in the concentration of intra and extracellular ions. Okay, now who can tell me the concentration of Na plus and K plus inside and outside? Do you remember? One chart was given where I have
Yes, ma'am.
huh,
Na plus are more in outside and K
yes,
plus are more in inside.
very good, very good. So it is less inside and at outside Na plus is more, whereas K plus is less outside and K plus is more inside. Very good, good. So this is what is written over here also. So in neurons, K plus and organic anions are typically found at higher concentration within the cell than outside, whereas Na plus and Cl minus are found in higher concentration outside the cell. So we'll just go slowly, okay, and then we are building up the action potential. So now till now this is clear that the resting potential is -70 millivolt, and that time your Na K plus ATP is closed. Now, how do you calculate or how do you measure the resting potential? It is with the technique called patch clamp technique. Okay, good. Okay, so this is how it just the diagram which shows that you have got the electrodes which records the measurement. Okay, now I have told you that the values are negative over here. Why? And here it is positive more. Why? Can you tell me? So remember I have mentioned about the negatively charged proteins, peripheral proteins. Okay, so mainly this negative charge is because of those negatively charged peripheral proteins. Okay, plus we have got Na plus in high concentration outside. Now when you attach the voltmeter and the electrodes are in connection with the cell membrane, what you get is around minus millivolt. Okay, this is just a cartoon representation. Actually, I'll take later. Now, the difference in concentrations provide a concentration gradient. It provides a concentration gradient of ions to flow down when the channels are open. Okay, so this is a scenario where it is given that Na plus is more outside and K plus is more inside. Okay, now these concentration gradients are maintained by the action of Na plus K plus ATPase by active transport, which in turn allow membrane potential to be maintained. So as far as now we know all this thing. Okay, so this is clear to us. Now what is happening? Let's see. Now how it is generated, the action potential is generated. So this is we are going to learn now. So during the resting state, the membrane potential arises because the membrane is permeable to K plus. Okay, and this action potential is happening at the axon hillock as a result of depolarization. So what do you mean by depolarization over here? It is when the electrical stimulus has reached the neuron and it results in the opening of your sodium and it results in the passage of the sodium ions. Okay, so during depolarization, voltage gated sodium ion channels open due to electrical stimulus. So are we talking about here Na plus K plus ATPase? No. We are talking about voltage gated sodium channel only. Getting the point here? See, when we have on the plasma membrane, one is your Na plus K plus ATPase. Okay. Then what we have is extra Na plus voltage gated and extra K plus voltage gated. Clear? Is this clear? So it's not that only Na plus K plus ATPase is present. Along with these active transport, I also have voltage gated Na plus channel and voltage gated K plus channel. Okay. So now, so what will happen? So once the sodium ion channels are open, it causes depolarization. So what will happen then? The sodium rushes back into the cell. The positive sodium ions raise the charge inside the cell from negative to positive. So then what will happen when so much of Na plus will go inside? Your minus 70 millivolt will become now less negative. Okay. So that is around minus 55 or minus 50 or something like that. So what is happening? This process is called depolarization. Now, is it clear? This much is clear what had happened till now? We were at rest.
Ma'am,
Huh?
once, ma'am. Once again.
Okay, once again. Okay. So what had happened is, so we know that at resting potential it was minus 70 millivolt. Okay. Now a impulse has come from another neuron. Impulse has come. Now what that impulse means? It is nothing but the passage of the ions. Okay. So what is happening? So depolarization means, I hope this is clear, that along with the Na plus K plus ATPase, additional voltage gated Na plus is there and additional voltage gated K plus channels are there. Okay. Now what had happened? Now what do you mean by axon hillock? So you have got the, suppose this is your cell. So at this junction, the dendrites of one and the axon of the other. So there the transfer of voltage happens. Okay. So at this junction, depolarization is happening. So depolarization means what? It means that from the plasma membrane, the voltage gated Na plus channel are open. Okay. So from outside to inside, your Na plus is coming. This much clear? So already the membrane potential was minus 70. When excess of Na plus will come, what will happen? This becomes less negative.
Yes, ma'am.
Okay. This thing is called depolarization. Okay. So as the sodium rushes back into the cell, the positive sodium ion raise the charge inside the cell from negative to positive. So it means what? From minus 70 to minus 50 MV it has become. So it is becoming less negative. Now what will happen? So since the concentration gradient is so high, what happens is that it keeps on increasing. That is, so many Na plus will come inside. What will happen then? The minus 50 will become to plus 30 MB. Okay. So it takes time for your channel to close. By that time, so much of ions will come inside. So that's why I divided each part into separate slide, because it's a very small topic, but confusing. Now, if a threshold is reached, an action potential is produced. Now, what has had happened now? So since Na plus has come inside and there is a change, depolarization happened. It means what? Action potential is generated. Okay, so if you see the graph, it becomes like this. Now, whatever has gone up will have to come down in order to come down to their normal state. And what is the normal state in the case of neuron? It is the resting potential, right? So this portion is called depolarization. Now coming back is called repolarization. Now, once the cell has been depolarized, the voltage gated sodium ion channels will now close. So what is the threshold? It has reached over here in the graph. If it has reached the maximum, that is called reaching the threshold of the neuron. Beyond this, the signal will not go. That is the capacity of the neuron to go this much. Okay, that is the called threshold. Now, once it has reached the threshold, the sodium channels will close. The raised positive charge inside the cell causes potassium channel to open now. So K plus ions now move down the electrochemical gradient out of the cell. Okay. So what had happened till now? Sodium, voltage gated sodium channels were open. It came inside. The negative potential has become positive. Now this change in the potential causes your K plus channel to open. Now what will happen? K plus from inside to outside it will go. Okay. So from your plasma membrane, now the K plus channels are there. From inside to outside, it is going outwards. Okay. As the K plus move out of the cell membrane, the potential falls and starts to approach the resting potential. Now it's clear. It means what? Because of K plus, repolarization is happening. Is it clear?
Yes, ma'am.
Okay. See, that's why I'm going slowly. Okay. Now, So now repolarization. So repolarization overshoots the resting membrane potential, making the membrane potential more negative. So what does it mean? See, it's just like the inertia. So what do you mean by inertia? If you're running, all of a sudden, if you stop, what happens? Your upper body moves forward, right? Because all of a sudden you have stopped, but your upper body was still in motion. In the same way, when K plus channels are there, okay. So when it is coming down, it means what? That from threshold it is coming down, it has to reach till the resting potential. One second, I'll show the graph. So it has reached over here. Now it has to come down, right? So it is coming down. But the process is like, under this threshold. So what is happening is overshoots the resting membrane potential. It means what? So many K plus from inside to outside it has gone now. So what had happened then? It goes beyond the resting potential. Getting the point? Now, this is called hyperpolarization. Depolarization, repolarization, if it goes beyond resting potential, it is becoming hyperpolarized. So hyperpolarized means now none of your neuron can get another threshold. None of your neuron can get another impulse. Is it clear this much?
Yes, ma'am.
It means that it is just a lag phase from becoming resting potential, just so that all the channels, can get adapted. So your sodium should get close, K plus should get close. Now, so it is important to note that Na plus K plus is not involved in repolarization process following an action potential. So till now we were talking about the voltage gated channels. Okay. Now from here to go back to resting potential, we need your Na plus K plus ATPase channel to become normal. Now you're getting. So first, because of Na plus, we have gone top, reached the threshold. Okay, sodium channels are closed, K plus channels are open, it is coming down, but it overshoots the resting potential. So it goes beyond that. Now over there, what happens? Your neurons becomes inactive. The channels become inactive. Okay, and that is the time at which a second threshold cannot come, a second impulse cannot come, unless until the first has been transferred from one to another. So from this, we also understand that the action potential is unidirectional. It means that unless until one signal has been passed to the another, it cannot take the another one. So from one cell to another, another to third one to fourth one. Okay, and you cannot have the back signal. It has to go in one direction only. Okay, so this is how your graph will look like. Now, every action potential is followed by a refractory period. So that overshooting the resting potential I was talking about is called refractory period. Okay, like this. So this portion is called refractory period. This period can be further divided into two: absolute and the relative. Please remember, these are important. This slide is important. Now, this period occurs as once the sodium channels close after an AP. AP means action potential. They enter an inactive state during which they cannot be reopened regardless of the membrane potential. So this we know, right? So this is called absolute refractory period. It means that once the action potential reaches its threshold, your Na plus channels are not open. So they get closed. Okay. So even if you give another threshold, the Na plus will remain closed only. Okay. Next, okay, slowly the sodium channels, one second. Okay, slowly the sodium channels come out of the inactivation, and then it is called relative refractory period. Okay, during which the neuron can be excited with another stimuli stronger than the already which has initiated the AP. Now, if you see the diagram over here, so till now what we were discussing, it's shown over here. So we have taken your minus 70 as your resting potential. Now, a stimulus is given, a threshold is reached, and it undergoes the depolarization. Correct? Then, this is when your Na plus voltage gated, Na plus are open. Then during repolarization, it is your K plus which is acting. Then there is a overshooting period, overshooting the resting phase. It is called refractive period, during which none of the other threshold can be taken or acted upon. And with the help of Na plus, K plus, ATPase, it goes back to its resting state. Okay, so it means what? One action potential has been generated and pass on to the next. Okay, clear?
Yes, ma'am.
Now propagation. So propagation means we are talking about that it from one neuron to another neuron it will go. Okay, so action potentials are propagated along the axons of neurons via local currents. So what local currents it is talking about? The flow of following depolarization results in depolarization of the adjacent axonal membrane, where this reaches the threshold and further action potentials are generated. So it just means that if you have got one cell over here, another cell over here, so whatever is happening over here, when it goes back to its resting potential, by the time it gives the action potential to the another one. Okay, so when this is under the repolarization state, by that time this undergoes the depolarization. The areas of membrane that have recently depolarized will not depolarize again due to the refractive period. Okay, so this means that the action potential will travel only in one direction. So just now we have discussed this. The local currents would eventually decrease in charge until the threshold is no longer reached. The distance that this would take depends on the membrane capacitance and membrane resistance. So do you know that in physics we have learned about the capacitors and resistors? Can anyone tell me what is the meaning? What does a capacitor do and what does a resistor do? See, I told you to do bio we need physics, chemistry, right? It is the same meaning over here also. So capacitance means ability to store charge. Okay. So here, one second. Sorry. So membrane capacitance, it is the ability to store charge. So if you have lower capacitance, it results in greater distance before threshold is no longer reached. So what you have to understand is, oh, time is getting up. What you have to understand over here is ability to store charge, less the capacitance, more distance it will go. Okay, the threshold, so that the action potential will be going for further distance. Similarly, resistance depends on the number of ion channels open. The lower the number, the more channels are open. It means what? It is showing less resistance. Less resistance means more number of your ion channels are open. A higher membrane resistance results in greater distance before threshold is no longer reached. Okay, this is the same concept we use for in physics also. Now, see the animation over here. So you can see how nicely the threshold has been passed. So you can see the Na plus is coming, then the channel gets closed, and then when this is getting closed, your K plus is getting outside. Okay, so it is the resting and the rising phase in your membrane happening, and similarly you can see how the graph is going. So resting means downwards only. The moment Na plus comes inside, you can see the threshold, it is rising, 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 and when your K plus is going, it is falling. Hyperpolarization. Can you follow the graph also as well? You can see that they have mentioned two figures over here, and both are different. Okay, this is of one cell, this is of second cell. So if you see, first it is having the Na plus, so that time it was your depolarization was happening, and then when it is going rising phase, it is falling. By that time, here it is depolarization, and then again it is rising. Okay. Yes. Now we have known about the myelinated axons. So I hope everyone knows what is myelinated axons. So on your cell, we have got the myelin sheath like this. Do you remember we have studied in our lower standards? Then we have got
Yes,
the
ma'am.
dendrites. Yes. So axons are there. Then we have got the myelin sheaths, right? And what are these called? The gaps? Anyone? Anyone? These are called?
Non-myelin.
Yes. Notes of Van Weir. Okay, it's just the what to say, it's just the revision only. So in order to allow rapid conduction of electrical signals through a neuron and make them more energy efficient, certain neuronal axons are covered by myelin sheath. Okay, so they actually help in the insulation. It improves the conduction by increasing the membrane resistance and decreasing the capacitance. So remember, more the resistance, more conduction will be there, and less the capacitance is needed. Okay. So there are periodic gaps along the myelinate axons where there is no myelin. Okay, and it is exposed. These are called nodes of Ranvier. Just now we have discussed. Now, myelinated sections of the axon, they don't have voltage gated ion channels. Okay, so there is high density of ion channels in the nodes of Ranvier. So wherever myelin sheath is present, there is no ion channels, whereas wherever nodes of Ranvier are present, it is having high density of high density of ion channels. Now, for this reason, potential can only occur at the nodes. So this we know. Now, the myelin sheath acts as good insulator, and so that the action potential is able to propagate along the neuron at a higher rate than it would be possible in the unmyelinated neuron. So what exactly is happening when this when the myelinated neurons are there? See, if you have like this, okay, if you have got the myelin sheath over here, then what happens? I hope you remember this. In school and all we have studied. So the conduction will be jumping. The impulse will be jumping. So from here to here it will be jumping. Then from here to here. Then from here to here. So the impulse will be transferred very fast. Otherwise, if suppose myelin sheath was here. One sec. Okay. If suppose myelin sheaths are not there, okay, and then what will happen? It has to pass like this. To each time it has to pass like this. So it will take more time. Okay, and this jumping of the electrical impulse from one node of Ranvier to the other is called saltatory conduction. This we have studied like in our lower standards. So the electrical signals are rapidly conducted from one node to the other, one node to the next, where it causes depolarization of the membrane above the threshold and initiates the another potential, action potential, which is conducted to the next. In this manner, an action potential is rapidly conducted down a neuron. Okay. So is this clear to you? Like how it is happening? If myelin sheaths are there, so the impulse will be jumping from one node of Ranvier to the another. So in this way, within few jumps, it will be passed from one neuron to the another. If it is non-myelinated, then it will take small steps. Okay, and it will take time. Now see over here. See the figure. So in the figure, oh, one second. Yes. So in the figure, you see that you have got the open ion channels, closed ion channels are there. Okay. Now what is happening? Sorry. Whenever there is an impulse transfer, it is going to jump from one to another. And you can see the gap is very small. Okay. So the transfer and the jumping will be very fast. And then this is called the saltatory conduction of action potential. Okay. So this is just the, what do you say, the summary of what we have learned today. Now, when they talk about the synapse, it means that the axon tails will be having some neuromuscular junction or somewhere. So depending on the signal, it is going to have the activity. Okay. Is it clear? So far, so good? Yeah?
Yes, ma'am.
Okay. So, okay. So now about the clinical relevance of this action potential. So this is one of the disorder we are going to talk about now. It's about the multiple sclerosis. I'm sure you must have heard also. And till now there is no cure for this, but this can be, can be controlled, but there is no cure for this. So multiple sclerosis is an acquired chronic autoimmune disorder affecting the CNS. So what do you mean by autoimmune? What do you mean by autoimmune? Yes, Meher?
Which is against our immune system.
No, it means that your immune system
Immunity is against us.
yes. So your immune system or the immune cells are attacking self cells or own cells. Yes. Okay. So in this case of multiple sclerosis, it is attacking your neuronal cells. Okay. So it results in demyelination. So it means what? None of your neurons will have the myelin sheath. Gliosis. What is gliosis? So your glial cells are there. It will be killed and eventually into the neuronal damage. Okay. So common presentation of the disease are optic neuritis. That means you are not able to visualize. Then transverse myelitis, that is your neurons are not functioning, and cerebral related symptoms such as ataxia. So ataxia means where you have seen that, sometimes there are continuous motor function is there or sometimes there is a paralysis. So once your neuron is gone, neuronal damage is there, you cannot predict what kind of loss or gain of function will happen. Okay, because everything we need, it has to be in our control. If I want my fist to be like this, I want it like this. But there are people who are not under control, so they will have this movement continuously, or sometimes they cannot move itself. So it's like they look normal, but their body doesn't respond. Okay, so a kind of paralysis happens. Okay, so this is the multiple sclerosis, and you can see what are the symptoms. So like half of the things even I don't know the meaning also, but yeah, that's okay. But what we understand from this is that fatigue, it's the tiredness, cognitive impairment, very difficult for the person to understand what exactly is happening, depression, anxiety, and unstable mood. Now visual nystagmus, optic neuritis, diplopia, it means that sometimes you see the double vision. Okay, so many things are there. Then even you're not able to control the urinary retention also. So it's because, see, even your bladder is under your control. If you want to go for urination, it is in your control to control the bladder contraction or relaxation. So when the neurons are not proper, what will happen? You cannot hold it. So whenever the urine comes, you cannot hold the bladder, and then the person is actually, there is a loss of the urine then. Okay, yes. Now, so there are three main patterns of disease. This is what is important. So what are those patterns? Relapsive remitting, it means that it faces the episodes of remission during which no symptoms are present. So it is the initial stage at which the person loses its neurons. It's losing its neurons, and initially you don't see the symptoms. See, because what happens is, if for 2-3 days I say that I keep forgetting things, okay, so that looks normal because your mind is stuck with so many things, so much of studies and so much of house course works are there, kids, everything, office, job. So if you forget 2-3 things, that looks normal, right? So it is very difficult to diagnose that a person, for example, like me, I have got lots of work to do. If I forget 2-3 things, does it mean that you can call me a multiple sclerosis person? No, because this happens commonly. But now if this is happening routinely, like every day completely I'm forgetting a particular thing and not able to remember also, like in order to say also that yes, I forgot. Okay, because for that person, the person who is having this problem, for them that thing doesn't exist at all. Like me going to office, I might forget completely, and if suppose someone comes and tells me, why didn't you go to office, I won't even remember whether I was going to office before or not, right? So because that particular portion of your brain, that memory has been lost. Okay, now coming to the secondary phase. So initially it is of the relapsing remitting pattern. However, at some point, the disease course changes and the neurological function gradually worsens. It means what? The complete loss of memory or the complete loss of neurological activity. Okay, there are certain things that I can, write very nicely, but after that damage of neurological damage has been done, now even I can't hold the pen. So because of this, what is happening? A permanent damage has happened. Okay, so this is the second stage. Now, what is the third stage? Now, primary progressive. Primary progressive means after the onset of the disease, there is a steady progression and worsening of the disease. It means what? Now it cannot be cured. So if it is good to diagnose at the first or the second stage, it can be cured or it can be controlled by giving certain medications. For example, like high doses of corticosteroids. Now, why we use corticosteroids? Because these are the only, they come under the steroids, steroidal drugs, because they are the only which can pass your blood-brain barrier, your brain, right? Blood-brain barrier. They are the only which can cross. Other drugs cannot. It will be then detoxified. Okay. So these are the only things which can cross your brain. And the problem with this steroids is that it affects your liver. So eventually with one disease, you start getting the another disease. Okay. Is it clear? Clear to everyone, Ramya? Sandhya? Okay. Thank you guys for listening to this. I hope you understood all.